Hello students, very warm welcome to you all for e-learning of pharmacology and toxicology. Okay, so today we will start with our very first chapter of your syllabus. So it's an introductory part, okay? An introduction and scope of pharmacology. Okay, as your name of subject include what pharmacology and toxicology. So we need to find out its importance and its use in pharmacy. Okay, so let's start with our introductory part. First we'll see introduction. Okay, introduction in terms of its definition and all points of view. Okay, so pharmacology means the study of drug, their sources, their nature and their properties. And pharmacology is the study of body's reaction to the drug. Okay, how our body can react with that particular administered drug is nothing but the pharmacology study. Okay, so past, we'll see the past history. Okay, next materia medica and pharmacy. Okay, what is meant by materia medica? Okay, so we'll discuss first what is meant by materia medica. It is the branch of medical science concerned with the study of drugs used in treatment of disease. Okay, it include pharmacology, clinical pharmacology and history and physical and chemical properties of that particular drug and the drugs used in treatment of diseases. Okay, सो आपलं जसं आय पी असतं ओके इंडियन फार्माकोपिया लाईकवाईज ॲट अँशियंट टाइम्स देवर मटेरिया मेडिका which include herbal drug, their sources and all important matter related with that particular drugs. Okay, present academics, presently in pharmacology is having huge scope in academics, also research in industries and in special domains. Okay, like forensic pharmacy and all. Okay, in academics, why it is included in academics or your syllabus? Because we are a pharmacy students, right? So we need to see what it contain, right? What is mean by pharmacology, what it contain. Previously it is only included in medical students or students with mbbs and all degrees. Okay, but now it is included in pharmacy also because we need to study thoroughly for formulation of new kind of drugs. Okay, so next is in research. It is very important point because wherever we are talking about research, that time we need to emphasize this pharmacology word. Okay, pharmacology means what does body do to that drug, right? So in research there are various new aspects by which we can reveal various new drugs. Okay, in industries, it is included in industries also, pharma industry and it industries also. Have you heard about pv pharmacovigilance? What is meant by pharmacovigilance? Pharmacovigilance is the study of side effects or toxic effect of drug. Okay, so it is also included in industries. Special domains means pharmacology study is included in forensic pharmacy and labs also. Okay, for criminal purposes and all other aspects. In future, what will be the futuristic approach of this pharmacology? We'll discuss over here. First, proteomics. For proteomics means what? It is large scale study of proteins. Okay, the study of proteins in large scale. Proteins are vital part of living organisms, right? With many functions, protein plays very important role in our body, where it will help for building our body bones, growth and all the factors, right? The proteome is the entire set of proteins that is produced or modified by an organism or system. Proteomics has enabled the identification of ever increasing number of proteins. Okay, so next is pharmacogenomics and pharmacogenetics. Okay, These both terms are very different, right? Pharmacogenetic means it refers to how variation in one single gene influences the response to single drug, right? It is only dealt with one single gene. Okay. And pharmacogenomics is broader term, right? So it include complete genes, or which studies how all the genes or genome can influence responses to drugs. Okay. Don't get confused between two words or definitions. Pharmacogenetics only deals with how variation in one single gene influences response to single drug. And pharmacogenomics means it is broader term, which which relate or which studies how all genes can influence responses to drugs. Okay. So, and next is nanomedicine. Okay. Nanomedicine is a targeted drug delivery system kind of stuff, and it is an interdisciplinary field of science. Okay. Bioinformatics combines biology, computer science, again IT or information engineering, mathematics also, and statistics to analyze and interpret the biological data. So here nanomedicine includes all the various aspects also. Okay. It is a branch of medicine that applies the knowledge and tools of nanotechnology. Okay. नॅनोटेक्नॉलॉजीचा यूज आता खूप होतोय. ओके. फॉर प्रिव्हेंशन अँड ट्रीटमेंट ऑफ डिसीजेस. Nanomedicine involves the use of nanoscale materials like biocompatible nanoparticles and nanobots, okay, for diagnosis, delivery, sensing, or actuation purposes in living organism. This is all about overview of pharmacology in various aspects like past, present, and future of pharmacology. Okay. Next, we'll start with introduction to pharmacology. First, we'll discuss the definition. Okay. Study of substances that interact with living system. Living system means what? Our body. Okay. So study of substances means what here? The drug substance. Okay. That interact with our living system or our body through chemical processes. So what are those chemical processes? That chemical processes are receptor and drug binding likewise, or by binding to regulatory molecules and activating that receptor molecule, or inhibiting normal body processes. Okay. This is very simple definition of pharmacology. Okay. Or the study of drugs, their sources, their natures, and their properties. Okay. So pharmacology is the study of body's reaction to drugs, how our body can react with administered foreign chemical entity, or we called it as drug. Okay. So got cleared this concept. So we'll move towards scope of it. An area in which something acts or operates or has power or control. Okay. So it covers all the area, all acts. Okay. We'll discuss one by one over here. First of all, we'll discuss some historical part behind this pharmacological development. Rudolf Buchem, first laboratory for drug research, who have invented first laboratory for drug research. Okay. Investigation of drug is a task of pharmacologist. They have they told us that invention of drug is a task of pharmacologist. Next scientist is Oswald Schamberg. Okay. He is father of modern pharmacology. Sorry. Oswald Schamberg. Okay. He was the father of modern pharmacology, and he was also related to first general in pharmacology. Okay. So next part is materia medica. In this particular uh, in this particular slide, I have shown you how it looks. This is used as reference book in ancient times by that peoples. Okay. So here they have written as materia medica americana. So this is the book which was used by American peoples at ancient times. Regni vegetables. right? So what it contains? Book containing names of herbs and medicine prepared from it. Okay, it contains names of various kind of drugs, medicinal drugs rather, and medicines prepared from that particular drug, or we simply called it as homeopathic or ayurvedic, what we can say over here, compilation of drugs, right? Dioscorides, he was a father, he is a father of Medica. Okay, methods for purifying active agents from crude materials were absent, hence real understanding of mechanism of action was absent or not known by that peoples. Purification process, isolation process was not written over there, so mechanism also was not known by that peoples also. So it is branch of medical science concerned with study of drugs used in treatment of diseases, includes pharmacology, clinical pharmacology, and history and physical and chemical properties of drugs, and the drugs used in treatment of various kinds of diseases. So we'll discuss our profession over here, pharmacy. No need to talk about this pharmacy, because pharmacy itself is a very huge branch, right? Its importance we all know. Science and technique of preparing and dispensing drugs. Okay, preparing and dispensing, we deal directly with preparation of drug, and also dispensing means selling of that drug to consumers or patients, right? Includes collection, identification, purification, isolation process, synthesis, standardization, and quality control of medicinal substances. Okay, so here first, they have included, or it include, pharmacies include single drugs collection method, identification, various chemical and physical identification tests, purification mechanisms, isolation techniques, synthesis, chemical synthesis, lab scale synthesis, likewise standardization. By modern analytical techniques we can standardize that particular new chemical entity, and quality control of medicinal substances. Okay, next is pharmacology is an essential component in study of pharmacy. Why this is so? Why pharmacology is very important? When I was in first year of B Farm, I only thought, or I my perception was like, okay, pharmacy is equal to pharmaceutics, because in pharmaceutics subject only we can um, prepare tablet, capsule, what we can say, cosmetics, that shampoos, lipstick, and all. Why there is need to study our human body? Right, na? I was not in favor of pharmacology at that time, but uh, however, the days going on, so my interest will develop in pharmacology so vigorously. Okay, and nowadays pharmacology is my very favorite subject, because it shows how our body react with that molecule, how drug response, or how receptor and drug will bind with each other, lock and key model, various receptors. They are very interesting part of our pharmacology. Okay, so next is universe of pharmacology. Okay, in this we will discuss what point it covered. Okay, research point, academics, industries, these are the special domains of pharmacology, as we had discussed earlier. Okay, so next is research, stages of drug development. Okay, in research there are three stages of drug development. Whenever we are watching news regarding any new invention regarding chemical entity, or regarding new drug development, at that time we don't know the history behind that, or we don't know the background behind that new drug development process. So here we can study how new drug will discovered by scientists. Okay, so very first step was drug discovery phase. Second phase after drug discovery, pre preclinical phase was is there, and after preclinical phase they'll push to clinical trial phase. Okay, so first we'll see drug discovery phase. Okay, there are four methods by of drug discovery. So first is random screening. Randomly we can discover any kind of drug molecule or screen any kind of drug molecule. Okay, another one is serendipity. Here serendipity means what? Accidental drug discovery. Okay, or happy observation by chance, huh? Not by we every time we are saying this particular thing ki, yes, good luck, all the best, and all. But what is meant by luck? Here we can say ki accidental drug discovery. Okay, we are going for something else, but we get something other. Okay, likewise, serendipity in drug discovery implies finding of one thing while looking for something another else. Okay, this was the case in 6 of the 12 serendipitous discoveries reviewed in this paper. Okay, so example of various drugs which are invented by this method or serendipity is or aniline, purple, penicillin, lysergic acid, and meprobamate. Okay, chlorpromazine and also imipramine. These drugs are discovered by serendipity. Okay, so a scientist was go for another version or another drug, but they were luckily got this drugs. Okay, so next is rational drug designing, and last one, designing of a prodrug or active metabolite. What is active metabolite? Prodrug means what? Okay, जेव्हा तो ड्रग आपल्या बॉडीच्या बाहेर असतो तेव्हा तो इनॅक्टिव्ह फॉर्मॅटमध्ये असतो, बट वेन एव्हर इट गोज इन टू आवर बॉडी दॅट टाइम इट विल गेट कन्वर्टेड इन टू active form of that drug, so that it will react with our body parts, our receptors, and it will show the pharmacological effect. Okay, to treat or to cure the ailment or disease. Okay, so so designing of a product or active metabolite as a drug. This is the fourth stage of drug discovery. Okay, so next stage is preclinical or experimental phase. Here we are dealing directly with animals. Okay, the aim of this preclinical or experimental phase is to satisfy all requirements that are needed before compound is considered fit to be tested in humans. Okay, this is very much important because our anatomy and physiology is very much similar with mice, monkeys, and alls, right? So we need to find the safety provisions of our drug in that particular uh, what we can say animals. Okay, if they'll show ki yes, that drug is safe, that time thereafter we can use this in human volunteers. Okay, it requires 1.5 to 2 years, that means 1.5 year to 2 years. Okay, out of 10,000 compounds screened, only 10 qualify for preclinical evaluation. See, the data was so strange that out of 10,000 compounds screened, only 10 qualified products for preclinical evaluation. So it also deals with effect of various pharmacological agents on different animal species. Okay, effect of various pharmacological agents or drugs on different animal species. Aims to find out the therapeutic agent suitable for human use. Okay, out of that 10 specified or optimized compounds, we need to find one single compound which is suitable for human use. Okay, next is study of toxicity of the drug. So after optimization from all that drugs, we need to study their toxicity or side effect study. Okay, so next is study the mechanism and site of action of drugs. Mechanism and site of action of drugs, how they behave or how they interact, or how the mechanism behind its effect we need to study over here. Okay, and site of action of drug, where it will be acted. So it is done by in vitro and in vivo study. In vitro means what? Outside the body, and in vivo means what? Inside. Okay, so inside the body means animal experiments. Okay, and in vitro means lab scale experiments. So in vitro study include receptor characterization, enzyme inhibition studies, and cytokine activities like various cytokine enzymes activity on particular drug. Okay, so next is clinical trial and third phase. Okay, it's very much important, guys. So here, systematic study of new drug in human subjects. Okay, so in second stage, or in experimental or preclinical stage, we will study our drug in healthy, or we can say in healthy animals, right? But after proving efficacy and safety margins on that particular animals, we need to study these drugs in humans also, right? So clinical trial includes the study of drug in human. Okay, so it includes various phases. From that first phase, okay, it include 25 to 100 individuals who are healthy one, and open label determines safe dose. Okay, it determines first safe dose of that particular drug. Okay, pharmacokinetic, what it mean? Okay, sometimes described as what the body does to a drug. Okay, our body, how it react with that drug, refers to movement of drug into or through and out of the body. Okay, the time course of its absorption, bioavailability into bloodstream, also its distribution throughout your body parts, metabolism and excretion it include. So ADME म्हणजेच काय की ॲब्सॉर्ब कसा होतो ड्रग, ॲब्सॉर्ब झाल्यानंतर डिस्ट्रीब्यूट कसा होतो थ्रू आउट आवर बॉडी पार्ट्स, अगेन मेटाबॉलिझम कसं होतं, जे कॉम्प्लेक्स मॉलिक्यूल तुम्ही इंजेस्ट केलेले आहेत त्यांचं मेटाबॉलिझम कसं होणार आहे, टू गेट अ व्हेरी सिम्पलेस्ट फॉर्म फॉर इलिमिनेशन ऑफ थ्रू बॉडी. सो it will study in pharmacokinetics. Okay, and predictable toxicity also. Okay, well known or predictable toxicity or side effects. Okay, phase 2 includes patients with target disease, right? Open label means what? Healthy individuals who are not having any kind of history of that particular disease for which we can develop a drug. Okay, so phase 2 include a patient with targeted disease, right? So it determine efficacy of that drug. Efficacy means what? How the drug is effective. Okay, so definitive endpoint. Okay, next is early phase 2. It will create a study on 200 patients, single blind. Okay, single blind means what? Blind means what? Ki they are aware of what is going on with that, what's going on with that particular patient. Okay, blind means what? They are not known, or they are not having any kind of idea regarding what is happening with their lives, right? So this is all about your single trial, or phase 2 study. Okay, a clinical trial is called single blind when only one party is blinded, usually the participants. Okay, who are participated in that clinical trial are not knowing what kind of treatment they are getting. Okay, so this is called as single blind. म्हणजेच की जे पण हेल्दी व्हॉलेंटियर्स असतील किंवा पेशंट्स असतील, त्यांना माहिती नसतं की त्यांच्यावरती कोणत्या टाईपची, कोणत्या ड्रगची ट्रीटमेंट होते. त्याला आपण काय म्हणतो? सिंगल ब्लाइंड और वन ब्लाइंड. ओके, इफ बोथ participants and study staff are blinded, it is called as double blind study. Participants means what? We all, right? So the healthy volunteers or volunteers for clinical trial, and staff. Study staff means what? The doctors, physicians who are डुईंग क्लिनिकल ट्रायल. ओके, म्हणजे दोन्ही बाजू ज्या आहेत त्या जेव्हा ब्लाइंड असतात त्याला आपण काय म्हणतो? डबल ब्लाइंड स्टडी. बोथ आर नॉट नोईंग व्हॉट इज हॅपनिंग. ओके, म्हणजे डॉक्टरना पण माहीत नसतं की ट्रीटमेंट कोणती दिली जाते, अँड ऑल्सो पेशंट्सना किंवा व्हॉलेंटियर्सना पण माहिती नसते की कोणती ट्रीटमेंट, कोणत्या ड्रगची ट्रीटमेंट चालू आहे. ठीक आहे, त्याला आपण म्हणतो डबल ब्लाइंड स्टडी. डबल ब्लाइंड स्टडी is one in which neither the participants nor the experimenters know who is receiving a particular treatment. This procedure is utilized to prevent bias in research results. Okay, म्हणजे डबल ब्लाइंड स्टडी आपण का करतो? तर आपल्या रिसर्चमध्ये कुठेही बायस प्रोड्यूस होऊ नये म्हणून. ओके, so double blind studies are particularly useful for preventing bias due to demand characteristics or placebo effect. Okay, so very next point is late phase 2. It will carried out on 200 to 400 patients. This is also double blind. Okay, phase 3. This study is carried out in huge population, 1000 to 5000 and plus. Okay, large scale multi-center double blind. Okay, to further establish safety and efficacy of drug. These three phases take 5 to 6 years to develop a single drug molecule. सी, आतापर्यंत आपल्याला खूप इझी वाटत होतं की न्यू ड्रग येतोय बाजारामध्ये, इज लाईक येस हां सायंटिस्टनी इन्व्हेंट केला आहे ड्रग, बट बिहाइंड दॅट दे आर हॅविंग व्हेरी हार्ड what we can say efforts. Okay, was there, and the efforts taking time like 5 to 6 years. Okay, and after that they will file a new drug for NDA application for licensing. Okay, what is the NDA? NDA include enough information for FDA. FDA is regulatory authority, that means Food and Drug Administration, to determine whether the new drug is safe and effective or not. Okay, whether the drug's benefits overweight its risk or not, whether the proposed drug label or package insert is appropriate or not, that means the mislabeled cases, right, and whether the drug manufacturing standards are adequate or not. Okay, these all points were cross verified over there. Okay, next, phase 4, very last study, post licensing phase. After getting approval from FDA, it will go for post licensing phase 4. No fixed duration was there. Periodic safety update reports like PSUR is to be submitted. Okay, now what is meant by PSUR? PSUR is nothing but periodic safety update report, is a pharmacovigilance document. Pharmacovigilance includes side effect data or data of toxic effect, right? So pharmacovigilance document intended to provide an update of the worldwide safety experience of medicinal product to regulatory authorities at defined time points for post authorization. Okay, this is all about your clinical trial phases. There are 4 phases: phase 1, phase 2, phase 3 and phase 4. Okay, so these are very much important one. Next part is reverse pharmacology. What it means? First of all, we'll see the definition of it. Reverse pharmacology is the science of integrating documented clinical or experimental hits into leads by transdisciplinary explanatory studies and further developing these into drug candidates by experimental and clinical research. Okay, so examples of reverse pharmacology is Ralphie alkaloid in hypertension. Okay, we'll clear this concept. What is meant by reverse pharmacology? As it's the name itself suggests or shows us what it meant. Reverse means something opposite. Okay, it relates routine lab to clinic progress of discovery to clinic to lab. Okay, lab to clinic means what? Forward or only pharmacology, not reverse. But reverse pharmacology deals with clinic to lab practice. Okay, conventionally, first of all, we'll invent molecule, right? After that, that molecule will study on mice, rat, and all animals, right? After that, it will goes on trial on man, right? So but in reverse pharmacology, it will get reversed. Okay, so first of all, we'll trial on man, and next mice and molecule, then we'll determine the molecule. Okay, in the process, safety remains most important starting point, and efficacy becomes matter of validation only. Validation means what? To get validate only. Okay, so next, in next slide I have shown you how it happened in forward pharmacology approach. Functional activity in vivo or in vitro, example of extracts or natural products done on various humans and all. So from that we'll get a lead molecule, and next step is target identification. But in reverse pharmacology, identification of promising target of proteins was done by digital data, okay, or from isolated molecules, okay, and then screening for compounds interacting with the target protein was done, and from that the isolated molecule was the purified molecule was isolated, and functional activity in vivo was done. Okay, this is all about here reverse pharmacology. Okay, so up till this we have seen various important aspects in today's lecture. Next part we'll see in next lecture. Okay.